The protein synthesis. As to the question of what is it, protein synthesis is the process in which the cells make proteins. It can also be called as protein anabolism. It occurs in two stages, namely the transcription as the first and translation as the second. As to the question of what is it for? Now, aside from the fact that there are 20 amino acids in our body, all of this plays the role as a component of our hair and the nails, and even the muscles, and even the cellular component that is described in the middle, together with antibodies that serves as a defense mechanism of our body, the enzymes that serves as a catalyst in every biochemical process that's taking in our body, and as well as a part of the blood which runs through our veins and through our arteries, together with the brain and the nerves. Now, at the right side, it explains the 20 amino acids that can be found in our body. 9 of them is remarkably said to be cannot synthesized by our body. And 11 can be synthesized by our body. In a way, protein synthesis will complete and suffice the ever-changing and never-ending need of our body for protein. Transcription, the first step of protein synthesis. This is taken from the word transcribe and script. When you are asked to transcribe, then you have to print a form. And when we say script, then you must have a template or reference or a code. Now, according to Watson and Crick, they named transcription as the central dogma in biology. Watson and Crick are the pioneers in DNA, or they are the discoverers of the DNA, where they describe transcription as DNA makes the RNA and RNA makes a protein as a central dogma. This is a process by which DNA information is copied into a new molecules of mRNA as a template or reference. As to the question of who copies the template, let us take a look and a listen at the clip below. As we recall cell, from within the cell, at the centermost is the nucleus, the center of control and command. And within the nucleus are 23 pairs of chromosomes that contains the long strand DNA packaged within a protein histone. Within the DNA are sections of the gene. This dictates the instruction for protein synthesis. As to the question, where is it happening? As previously been displayed, transcription is happening within the cell, in the innermost nucleus, at the chromosomes where the DNA and the gene can be found together with the mRNA. Transcription chain initiation. As to the question, how will it happen and what initiates the process, let's take a look and listen. Transcription begins as it is initiated by an enzyme RNA polymerase, that big bulky brown structure at the left. It will attach itself to a gene and unzips the DNA. It will move the strand of copied pre-bases template and will form the DNA code that will dictate the sequence of base formation from the RNA. This process is called chain initiation in transcription, and this will be followed by chain elongation. The chain termination of the transcription. As to the question of what is the final step of transcription, before the mRNA can be used as a template, it must be processed. And this is done by removing and adding the parts of mRNA called the splicing reaction. It happens before it exits the nucleus for translation. The translation. As to the question of what happens during translation, translation is the second step. Decoding of mRNA into protein happens. The transfer RNA carries amino acid from the cytoplasm to the ribosome. What are the organelles involved, and how will it happen? The mRNA will move out of the nucleus to the cytoplasm. And protein factories in the cytoplasm called ribosomes will bind to the messenger RNA. The ribosomes reads the code in RNA to produce a chain of amino acid. As translation continues, the question may be, what is the role of the RNA in the cytoplasm? Each tRNA has 3 nucleotides that are complementary to the codon in mRNA, and each tRNA codes for different amino acid. How will it then create a polypeptide chain? A series of 3 adjacent bases in mRNA codes for amino acids called codon. mRNA reads 3 bases at a time. As each strip are read, the tRNA delivers a corresponding amino acid, adding to a growing chain of a polypeptide. What step will complete translation? Once the last amino acid has been added, the chain folds into a complex 3D shape to form a protein. And why do proteins fold? Proteins folding is the process by which a protein structure assumes its functional shape or conformation. By coiling and folding into a specific 3-dimensional shape, they are able to perform their biological function. Once again, we have reached the end of discussion. Let us assess and check your comprehension. You may pause the video for a good glance. Enjoy studying, and I hope this material is of help. All the best to you. That's all, folks. This has been your Nurse Radiologico Guro saying thank you for watching and enjoy studying.